the workshop on surveillance and data analytics of antimicrobial resistance in public health. I'm Aparna Balchandran, Program Coordinator at the NanoScience and Technology Consortium. Before we begin, I'd like to take a moment to introduce NSTC. Established in 2006, it's a leading edtech organization dedicated to promoting research and innovation in emerging fields such as nanotechnology, biotechnology, and artificial intelligence. Through our specialized training programs, interactive workshops, and virtual learning platforms, we aim to empower students, researchers, and professionals to upskill and contribute meaningfully to the global scientific and technological community. Now, coming to the today's session, this workshop focuses on how public health and research communities track, interpret, and use AMR data for surveillance, decision-making, and prevention strategies. Before we introduce our mentor, a quick note for participants. Please keep your microphones muted during the session. You can post your questions in the chat box and we will take question and answer at designated intervals at the end. The recording and e-certificate will be shared via your NSTC dashboard. Now it's my honor to introduce today's mentor, Ms. Aishwarya Arun. Ms. Aishwarya is a microbiologist affiliated with Dr. Rahul Patil Hospital and Research Institute, Maharashtra, India. She holds an MSc in her domain and has a strong research and practical experience in microbial susceptibility, antimicrobial activity, and sustainability-focused microbiology. She has published multiple research works in referred journals, and she also contributes to the scientific community as a reviewer for international journals, including Elsevier and Springer Nature, as an editor with an international publishing house. With her combined expertise in laboratory microbiology, research writing, and mentoring, she will guide us through a highly relevant and applied AMR surveillance workflow. Now, in the workshop, we will cover AMR surveillance frameworks and data sources, global AMR surveillance, WHO, GLASS, NARMS, and One Health initiatives, accessing and understanding WHO, GLASS, NCBI, pathogen detection dashboards, data retrieval, and format harmonization, metadata integration, pathogen geography, and resistance class. So without further delay, I now hand over my session to our mentor, Ms. Aishwarya Arun ma'am. Over to you.
Thank you so much, ma'am. Thank you for introducing me such nicely. I welcome you all participants in this workshop of AMR surveillance. I'm sharing my screen. Just 1 minute. Hope the screen is visible to all the participants.
This call is now being recorded. This call is now being transcribed. This call is now taking notes.
Please, ma'am, let me know if the screen is visible to all the participants or not.
Yes, it is visible now.
Okay, thank you so much. So today we are going to see about AMR surveillance frameworks and data sources. So in day 1, we are going to focus on understanding global frameworks and learning how to access real surveillance data. Well, before going ahead, what is this AMR? That is antimicrobial resistance. What is antimicrobial resistance? See, antimicrobial property is the property of an microorganism to have certain reaction with the antimicrobial drug. It may be chemical, it may be a gas itself. But what happens with antimicrobial resistance, that these microorganisms adapt certain resistance capacity to that antimicrobial drug. It does not resist to that drug, and what happens, they still survives even if it get contact with that chemical. So because of that, nowadays this AMR problem is being extremely increased worldwide. Global death count is increasing immensely. Why? Because we are adapting with many chemicals every day in our everyday life, like fertilizers, pesticides, and exactly the way we are adapting to these pesticides, fungicides, various chemicals, exactly the same microorganisms are also adapting to such kind of chemicals, and they are living very easily with in environment with them. So because of that, the disease control is reducing day by day. So what the work of a drug specialist, what is the work of microbiologist, what is the work of pharmacist over here is to have a proper AMR data, to analyze that data and generate certain drugs which are really worthful. And what ultimately result may be, the result should be to decrease the an AMR death count, antimicrobial resistant pathogenesis, and because of that the death count should be decreased. So that's why this AMR surveillance framework and data sources, such kind of workshops are nowadays very important. So I welcome you all once again. I welcome you all in this very good, very nice topic to be explored by me and you people. So let us go ahead. In this today's session, we are going to see global AMR surveillance frameworks. In that, global frameworks exist to track resistant trends, guide policy, and support clinical decision making across countries. Second is demo, which is accessing AMR dashboards. In that, this section introduce real world surveillance dashboard used by researchers and policymakers. Then hands on data retrieval and format harmonization. In this section, focus on working with real AMR datasets. And then fourth is the activity, that is metadata integration. Okay, before going ahead, I would like to tell you all that real hands-on and actual practical activity we will keep on last day. That I will include every day's activity section and I will keep it in the last day, and I will explain that every activity in thoroughly, how to access the database and how AMR dashboard works actually. So let us see today global AMR surveillance frameworks. So first, WHO GLASS, that is Global Antimicrobial Resistance and Use Surveillance System. So this is a GLASS system. This is coordinated by WHO itself. Why this system is working continuously? Because in medical field we consider the death by the pathogen, but we do not consider that the pathogen's nature to that particular drug, how it has reacted to that particular drug. So this AMR surveillance is mainly capitalized by glass country. Glass it's not a country, it's a association which is being organized, which is coordinated by WHO. And what every country has to do in it is they have to include their AMR data every year into it to maintain the country national records, to maintain the global records. So what it is, GLASS is a WHO coordinated global platform launched in 2015 to standardize AMR surveillance across countries. So, what is the purpose of it? Compare AMR trends between countries, then identifying emerging resistance and support evidence-based national policies. So, we all are very well aware about the biggest pandemic of coronavirus. So now corona has been thoroughly studied very well. We very well know about its origin, its genetic content, its gene sequence, how is actual the structure of that corona, how it is getting multiplied, how it is getting inherited. Everything we are very well know, we very well know about it. But how it is, because the scientist has worked on it and they have put forth a data that is being recorded worldwide, and so sitting everywhere the data is available to every person, and so that we can study it furthermore. So that is again about the glass framework itself, that maintaining the databases, maintaining the structural analysis, maintaining the national records, and maintaining the global records, and finally analyzing which microorganism is affecting more, which country has more AMR surveillance, which country has less AMR surveillance. Then the key feature is standardized case definitions, uniform laboratory methods, then common data reporting formats, and covers both AMR and antimicrobial consumptions. So that is the key feature of this AMR surveillance framework, WHO glass community. See, such kind of screen you will be that you will find on WHO's glass community. On the screen you will find every nation putting every year's data into it, that how AMR surveillance is working, which pathogen is working more, which pathogen is working less, about antimicrobial resistance, antimicrobial susceptibility, again consumption, all the things that every country is updating every year. Such kind of framework you will find on the screen of WHO's glass community. Again, here I have just I have included one of the another figure of WHO, which is showing all age rate of death attributable to and associated with bacterial antimicrobial resistance by GBD region, global boundary death region. Okay, so you can see that from western sub-Saharas Africa to the Australasia up to here included many countries, they have kept their death rates per 1 lakh of population. So here such kind of analysis every year that WHO's glass community generates. So this is for year 2019. Okay. Again, we are going to see every year's death count, every year's AMR surveillance rates and all in upcoming slide. Again, this is for 2019 that WHO has put forth that which bacteria is highly resistant to antimicrobial drug and is capable of having increased death count every year. So we very well found that Escherichia coli is having highest death count percentage. So what we can conclude over here is Escherichia coli is highly resistant to about 98% of the antimicrobial drugs and is capable of having largest death worldwide in GBD regions. So there are around many pathogens has put forth over here. You can see there are Pseudomonas aeruginosa as well. Then you can find Klebsiella also. You can find Staphylococcus aureus also. All the pathogens. And how much is their microbial resistance property, antimicrobial resistance property, and then its worldwide death count that is given in this data attributed by WHO's glass community. Now, which pathogens are highly monitored every year? First is E. coli, second is Klebsiella, third is Staphylococcus aureus, then fourth is Staphylococcus, pneumonia, and Salmonella species. So these 5 pathogens are highly studied every year because their infections are very emerging. Every person has infected with these pathogens definitely, and we usually take their drugs in our day-to-day life, and that's why the reason for main antimicrobial resistance is our day-to-day adaptation to various medicines, and those medicines like antibiotics, like fungicides, all these things we used to take it randomly, making our body incapable of taking those drugs and ultimately making those pathogens get adapted to such antimicrobial drugs and leading to the AMR, antimicrobial resistance property of that particular pathogen. Okay, data type collected. So which type of data types, which type of sources for the demonstration of this AMR is required? First is patients demographics such as age and sex, which is that patient, it is highly aged, or it is just a young one, or it is just a toddler, it is just a child, it is a even if it's a female or if it's a male. Then specimen type, that is blood or urine, pathogen identification, and antimicrobial susceptibility results. Such a type of data are very required. And in if you will find the AMR dashboard, then we have such columns over there, and into that columns we have to put whether we do not have to put the name of the patient directly, we just have to write their age, then the sex, whether it is female or male, then the type of specimen you have selected, whether it is urine or a blood or etc, or then what that urine result, what that blood specimen result you have obtained, that which type of pathogens are present into it, and after that how many antibiotics you have used to check the AMR result. All such kind of data we have to put into it, and then we have to include all those data in a national laboratory. Which is that national laboratory? It is a record maintenance laboratory of that glass. Every country has to do it every year. Why glass matters? Enable global comparability, then supports LMIC. What is this LMIC? It is low and middle income countries. In the World Health Organization, global antimicrobial resistance and use surveillance system participation. So because of this, glass has enabled every country to monitor their data and to enable themselves to put their national data into this glass framework. And because of this easiest making, many of the LMIC countries are participating into maintaining their records as well. So, last is forms the backbone of WHO AMR reports. So, none of the country is remaining back. Every country is proceeding into it, putting their data annually, maintaining the global records, and helping to the World Health Organization community to find which pathogen is emerging, which pathogen is being degraded. All those things are being calculated by them. Next is NARMS, that is National Antimicrobial Resistance Monitoring System. Well, what is this? The National Antimicrobial Resistance Monitoring System is a US public health surveillance system tracking antimicrobial resistance. So it is again US Organization of Public Health Surveillance, which is tracking antimicrobial resistance in enteric bacteria from humans, food animals, and retail meats. A collaborative efforts between CDC, we very well know it is a disease control agency, FDA, it is a Food and Drug Administration agency, and USDA, it is for especially for the agricultural resources. So, in combination of the CDC, FDA, and USDA, the NARMS collaborates their efforts in analyzing public health surveillance and for tracking, ultimately for tracking the antimicrobial resistance. It monitors resistance trends, identifies new resistance pattern, and provides data to guide policies, improves food safety, and promote judicious antibiotics use in human and veterinary medicine, operating under One Health approach. Okay, this is their slogan, One Health slogan, to connect human, animal, and environment health. Okay, in One Health, they have included humans, animals, and environment. Humans, animals, and environmental sciences into it for their for their health analyzing. How it is? Let us see it. Now, what is the purpose for it? NARMS is a US based surveillance system led by CDC, FDA, USDA. Then, their primary focus is to identify foodborne pathogens and resistance arising from animals, agricultures, and humans ultimately. Which pathogens are highly monitored under this? First is Salmonella, then Campylobacter, and then E. coli, and then Enterococcus. These 4 pathogens are highly identified, highly analyzed, examined under this NARMS norms. So, surveillance resources are humans, retail meats, food producing animals. So, these are three are the surveillance sources. From these sources, the pathogens are generally collected humans. Then retail meats. Retail meats we very well know retail meat is of animals, and food producing animals. Okay, such kind of things are such kind of sources are used for the surveillance pattern. Why NARMS is important? Okay, it tracks resistance transmission from animals to humans, informs food safety regulations, and helps regulate antibiotic use in livestock. So, must understand this concept that what is the main purpose of NARMS? They having a wide purpose of analyzing the pathogens. See, we very well know that many of the pathogens are contagious. They transfer from animals to humans, and even from the plants to the humans. So, to avoid such spread of pathogens and from them to avoid their microbial resistance capacity, this NARMS works. Okay, suppose any pathogen is already been resisted to the antimicrobial drug and it is affecting the plant, and it is being contagious to the human as well. So if that pathogen is already AMR, and if it secondarily it infects the human, it will not even it will definitely affect to the human also. It will not no even the human antimicrobial drug will is having capacity to reduce that pathogens surveillance. So that is what this NARMS is working for, completely eradicating that primary disease causing agent, whether it will be contagious or not, whether it is being transmitted from plants to the animals and again from animals to the humans. So their purpose is to widely study the disease causing pathogens and their AMR, antimicrobial resistance capacity, and to identify their pattern of resisting to the um, plant drugs, veterinary drugs, and human drugs, everything. So that is why the NARMS is important. Next is One Health AMR initiative. Okay, that is their slogan, One Health. What is this One Health? It includes humans, animals, and environment completely. Concept One Health recognizes that human health, animal health, and environmental health are interconnected. As I told you, nowadays we found that every day we are being our World Health Organization is releasing a data of newly generated viruses, newly generated pathogens. We are getting known to various bacteria every day. They are being identified. So we don't know that which disease is transmitted from where, what is their original position, what is which environment is their origin, whether it is originated from plants or from animals or from humans. We don't know them. So in One Health, it is their main motto is to identify the disease causing pathogen, to identify its origin, and then to identify a large number of AMR pattern. For their purpose is to eradicate that pathogen completely and to reduce its antimicrobial resistance pattern. One Health surveillance integrates human clinical data, veterinary and livestock data, and environmental data. So it integrates human clinical data, then veterinary and livestock data, and environmental data as well, like soil, water, and wastewater. Example of One Health data sources are hospital laboratories, definitely. We obtain the patients' data from hospitals, then veterinary diagnostic labs, and then wastewater surveillance and agricultural antibiotic uses data. So such are the sources, example of One Health data sources. Why One Health is critical. Okay. Many resistance organisms emerge in animal or environment. Resistance genes spread via food or water and human also provide early warning of emerging resistance. Okay, see that last line is very important. Which is that last line? This is provides early warning of emerging resistance. That is very important over here. Why? Because as I told earlier that many of the pathogens are they travel in three generations, like they travel in all three environments. Which three environments? Soil environment, plant environment, soil or plant, it may be plant environment, from plants to the animals, and from animals, that's those people who eat that animal, and that on all animal is already been infected, that pathogens get entered into the human body and to the human last. So from plant to the animals and from animals to the humans. So in this three cycles that pathogen is being traveled. So see, if a plant is being infected and we already having the data, AMR data, so we can conclude that those plants should not be eaten by the animals or not even eaten by the humans also. So that is what provides early warning of emerging resistance to what? To the antimicrobial resistance capacity of that pathogens, and ultimately protects the humans to get infected from. And that is ultimately the source and motto of this One Health AMR initiative. Next is demo accessing AMR dashboards. That I will show you this how to access the AMR dashboard. Okay, everybody must have if this everybody must have the WHO platform been installed in their phone or in their laptops or in their whatever the MacBook or everything, whatever it is, they must install the WHO framework into it, and into that WHO we will find a glass community. In that glass community, this AMR dashboard is there. Into that AMR dashboard we will find nationwide data. Also we can find every year's data over there, that how in which year that nation was having highly AMR resistance, AMR surveillance data, again death count as well, in which country, every year we can find out which country has highest death count because of this AMR, everything. A WHO glass dashboard, it is a what you can explore. First is country wise AMR prevalence, then resistance trend over time, then pathogen antibiotic combinations, then antimicrobial consumption data. So what you can find in this WHO dashboard is country wise AMR prevalence data. You can find over there country wise, there is a dashboard. You just have to put your country name over there and you can find it over. Then resistance trends over time, we have seen that such kind of resistance trend is there. What is this in showing? Escherichia coli is having highest AMR, highest resistance capacity, while this one, this last, this marginella species is having less, very less percentage of AMR, antimicrobial resistance, as compared to the E. coli. So you can find it over there as well. Then pathogen, then resistance trend over time, then pathogen antibiotic combination and antimicrobial consumption data. You can find over there how to use. First you have to open the WHO and into that glass community dashboard, and then select country or region, then choose a pathogen, then select an antibiotic and view the resistance percentage and trend. Okay, I will in brief I will show you this thing. We are going to see the same thing one more time during our last session. In that, the complete hands-on and complete activity are being taken on that day directly. Hold on, I'm sharing the screen. Just a minute. Hope the screen is visible to everyone. So this is a WHO data in which you can find welcome to the antimicrobial resistance AMR data portal. So you can find that there is antimicrobial resistance profile, antimicrobial use, infection prevention, surveillance of AMR, governance, awareness, education, laboratory diagnosis. So, into that laboratory diagnosis, you can find here is the nation. Okay, you just have to put your name of the nation and you can find their result ultimately. Every nation is included into it because they have participated in updating their data regularly. And suppose any nation do not provide their data, such nations are sent with the report that you have to submit your data into it, and that nation is urged to submit the data. So every year this record is being maintained. So you can find every country is included into it exactly like this. Then tracking AMR country self-assessment survey, TRACSS, that is capacity to perform antimicrobial susceptibility test for critically important bacteria. It is for year 2025 is not included yet. So rest of other data is been included over here. You can find such data over here. We microbiologists used to find out this data for our references to write during writing our articles, and this data is highly standardized. There is no internal community to maintain to who will distinguish with this data, who will do mess up with this data. So this is a standardized data. Every microbiologist, every medical associate used to find out this data for maintaining their articles perfection and highest reference range. So this is about the AMR dashboard. Like this, how to access the AMR dashboard? We have to select the country or region, then choose the pathogen, then select the antibiotic and view the resistance percentage of that country of that particular pathogen to that antibiotic. Again, find last is the educational value. Learn global resistance pattern. With the help of this AMR data, we can learn the global resistance pattern of that pathogen, understand the geographic variations, and compare high income versus low income region. Okay, we can compare the high income and low income region with the analyzing such data. NCBI Pathogen Detection Dashboard. NCBI stands for National Center for Biotechnology Information, is a key part of US National Library of Medicine NLM and the National Institute of Health NIH, providing last biomedical data, bioinformatics tool and resources like PubMed and GenBank. This is a gene bank for researchers globally. So, by the NCBI's records, by the data which has been published by NCBI, worldwide global scientists, global researchers which are working on pathogens, microorganisms, viruses, medical associates used to find this data from PubMed and GenBank, and they used to research globally with this data. NCBI pathogen detection dashboard. So what is it? A genomic surveillance platform using whole genome sequence. WGS, that is whole genome sequence. So with the help of this NCBI data, you can find the whole genome sequence of that particular pathogen. So see, there are certain standardized dashboards, like we have seen first AMR of glass dashboard, AMR dashboard, which is of that glass community and which is coordinated by WHO. This is NCBI. NCBI gives us the standardized data of genome surveillance platform using whole genome sequencing, and so that this standardized standardized data are having 0% errors. So we can use this. There is no mess up with any compromise, or there is no mess up with any error in that data. That's why it is the worldwide globally accepted data. Which are the data sources? Like clinical isolates, foodborne pathogens. Sources are like clinical isolates, then foodborne pathogens, then environmental samples. Such kind of sources are the data sources, sources which are used to collect the pathogens. What you can learn: how resistant how resistance gene spread, relationship between isolates, tracking outbreaks across countries. So into that particular pathogen, which is that antimicrobial resistance causing gene. Okay, we very well know that into bacteria there is a plasmid, and antimicrobial resistance gene is particularly placed into that plasmid. But whether it is of plasmid only, or it is any another nucleotide sequence, that is either of nucleoli or of that plasmid itself. So you can find it out easily with the help of this data put forth by NCBI, that what resistance gene is spread, relationship between isolates, and tracking outbreaks across countries. So see, if I have two similar species of the same pathogen, okay, I have two species of the same pathogen, but what is the difference? That first pathogen is resistant to another antibiotic and second pathogen is resistant to another antibiotic. Now you can analyze that same isolate of the pathogen by directly identifying that which is that resistant gene, and this resistant gene is specially resistance to which type of drug. What is this key features? SNP based phylogenetic clustering, resistant gene identification, outbreak detection. So these are the key features. A SNP based phylogenetic tree visualizes genetic relationship by mapping single nucleotide polymorphism, that is SNPs, single nucleotide polymorphism, to show evolutionary divergence with branch length reflecting the number of SNPs differences, helping to trace pathogen spread, understanding crop diversity, or map human ancestry using tools like SNP2Tree or KSNP to process vast genomic data and build powerful evolutionary hypotheses. And this is how it works. Okay. Now, with the help of this SNPs, what is this SNP? It is a single nucleotide polymorphisms. So, with the help of this data, what we can do is we can helping to trace pathogen spread, understand crop diversity, or map human ancestry using tool like SNP2Tree or KSNP to process vast genomic data and build. This is what? To build powerful evolutionary hypothesis. So ultimately the reason behind this NCBI's SNP data is to build powerful evolutionary hypothesis. And ultimately what we used to do is, we microbiologists used to analyze this NCBI data for this purpose: how resistance gene is spread, okay, and especially relationship between isolates. So for this main concern, we used to find out the NCBI's data, and which is the standardized data released by WHO and standardized US communities. Third is now hands on, that is data retrieval and format harmonization. What is this harmonization? Harmonization in AMR surveillance data is a crucial for comparing results across labs, regions, and sectors. Which are those sectors like humans, animals, and environment, enabling enabling unified global tracking, identifying resistance trend, informing one health policies, and accessing antibiotic uh, stewardship effectiveness, ensuring data quality, and supporting timely interventions against superbugs. So without it, data is inconsistent, making it impossible to see the big picture or develop effective coordinated strategies to fight the growing threat of antimicrobial resistance. So what is this saying? That it is a crucial data for comparing what results across the labs, region, and sectors. In this AMR harmonization, this data is retrieval and format harmonization is very important. And because of until and unless we compare our results across labs and regions and sector, we cannot find global tracking. Okay, this is important for tracking the data globally. That, as I told you earlier, that if I have to find that for the year 2025 the data is not generated yet, that will be definitely generated very soon. But if I were to find out that which pathogen was highly AMR antimicrobial resistant during the year 2025, then if I will put forward directly that pathogen into the glass communities AMR dashboard, then I will definitely get that pathogen's name. Now, second is what? I have to find out the percentage. So I will also find out that percentage over there. Now, third is I have to find the region, then sector. Now, region, I will definitely get this nation was highly affected by this pathogen during year 2025. And if I want to find the sector, that whether it is humans, animals, or environment, that data is also available over there. And how it is available now? Because of this data retrieval and format harmonization that is being monitored by various scientists which are working under this glass community. They are taking the records of every nations, they are updating their records. Scientists worldwide are working with various pathogens, working with their AMR surveillance capacity, their pattern, and including all their record into this glass communities AMR platform. And we, sitting in our home also, we can find this data, this globally analyzed standardized data, and we can use this data for further our articles, further our studies works, we can use it. Hands on data retrieval and format harmonization. Data, first is data retrieval. There are common data formats like CSV, JSON, API. What is the CSV? It is a spreadsheet friendly, widely used. JSON is a structured, machine readable. And then third is API. It is a programmatic data access. These are the common data formats that are generally used. Sources are GLASS downloadable reports, NCBI pathogen detection datasets, and National Surveillance Portal. So these are the sources for generating such data. Skill developed. So with the help of this downloading dataset, this such which skills are developed downloading datasets, understanding column definitions, and identifying missing or inconsistent data. So such kind of skills are developed into us. What is B? That is format harmonization. Why harmonization is needed? Different system uses different antibiotic name, different resistance breakpoint, different coding system. And so that to avoid this different analysis, to avoid this misconception or confusion, harmonization is very important to give unique name to that particular method, to that particular pathogen, to that particular system, antibiotic name, and unique name is given under this harmonization, and so that it is not a local name, it is a standardized name, and that's why harmonization is needed. Example of harmonization task: converting CIP, that is ciprofloxacin, is converted as CIP is converted as ciprofloxacin. So many of the scientists uses the short forms. So that short forms are properly written with the help of this harmonization task. Standardizing country codes, that is IND, India, that is in India form. Mapping resistance categories, SIR. Such kind of things are work of this harmonization task. Then what is the outcome of it? What is the outcome of it? Clean, comparable data set ready for analyzing and visualization. So, see, let us understand one more time data retrieval and format harmonization. Data retrieval, we can identify, we can generate the data through it, but for getting a proper data, this harmonization, format harmonization is very important. With the help of this harmonization, we format every data in a standardized data. We enlarge the short forms into proper names. So that is why the data retrieval and format harmonization is important. Fourth is a activity, that is, I will explain this in brief. This is the metadata integration. Okay, that this activities are for tomorrow also and day after tomorrow also. The same type of activities are there. That's why we are going to conduct this on the last day, hands on. Pathogen metadata. Pathogen metadata includes species name such as strain or sequence time. Resistance gene example is given over here: BLA Citex M or MEC-A. So it is elaborated with the name. The Citex M gene codes for CTXM type extended spectrum beta lactamase ESBLS. So this is what beta lactamase ESBLS extended spectrum name, that is written as the CTXM gene. Second is a MEC-A gene, is a bacterial gene primarily in Staphylococcus aureus MRSA that provides resistance to beta lactam antibiotics like methicillin by encoding PBP2A, penicillin binding protein 2A. So see, it is not completely written in that standardized data. Okay, how we have to convert it? We have to convert it in a short forms, but those short forms should be worldwide accepted, should be standardized, and should be easily available to all the person who, to avoid the confusion. So these standardized short forms are included into it. What is the importance? Identifies high risk clones, helps understand mechanism of resistance. So with the help of this data, we identifies high risk clones and helps to understand the mechanism of resistance. What is this B? That is geographic metadata. It includes country, region, hospital and laboratory. Why it matters? It tracks geographic spread, identifies regional hotspots, supports local interventions. So, because of this, what is that? It tracks geographic spreads, geographic spread of pathogen. It tracks it. Then it identifies regional hotspots. We very well know that during corona pandemic, the South Asian regions were the hotspot for its spread. So during that time, the news channels, the WHO reports everywhere, it was showing the hotspot regions to avoid the contamination of the coronavirus and to avoid its spread. So it identifies the regional hotspot. Then it supports local interventions. Ultimately supports the local interventions. And number C, it is a resistance class metadata. It includes antibiotic class, that is beta lactam, fluoroquinolones, and resistant mechanism like ESBL or carbapenemase. So such kind of resistance class metadata is there. Use cases are trend analysis, policy formulation, and antibiotic stewardship planning. So in these cases it is used, that is trend analysis, policy formulation, and antibiotic stewardship planning. For such kind of activity, this metadata integration is useful. So with the help of this today's session, I would like to conclude. One more time, I would revise some important parts of it. What is this global AMR surveillance framework? It is the global antimicrobial resistance and use surveillance system, that is a GLASS, which is coordinated by WHO itself. And under this GLASS, AMR surveillance dashboard is there. Into that dashboard, we can find the pathogens, region-wise pathogens, status-wise pathogens, nation-wise pathogens, every year's data set of death count, how much percentage of its antimicrobial resistance capacity is. We can find everything onto this AMR surveillance framework, and it is a globally recognized, it is a standardized data having minimum amount of errors into it. See, we can say minimum because see, no scientist is 100% perfect, 100% sure, but 99.9% can be easily said, because to avoid the we can say like a minimum percentage of error is there. Then such kind of data sets are generated by every nation and put forth into this AMR surveillance data board. Then this is the death count, every region wise, GBD regions death count is given over here of year 2019. Then this is the bacteria wise death count, which pathogen is highly affected, which pathogen is less affected. That bacteria wise death count is given over here. Which main pathogens are examined highly? Pathogens like E. coli, excuse me, sorry, E. coli, Klebsiella, Klebsiella pneumonia, Staphylococcus aureus, Staphylococcus pneumonia, and Salmonella species. Sorry. Data type collected. Which type of data type are usually used to collect the specimens, the pathogen species like blood, urine, and then the data which is collected from which type of patient, that is whether it is aged one, whether it is young one, whether it is a toddler, whether it is a child. Such kind of data is maintained, and then it is sent for the pathogen identification. Once the pathogen is identified, it is further used to test their antimicrobial susceptibility, and then this type of data is generated, and this data is put forth into GLASS, the AMR surveillance framework. And so why this glass matter is, it enables global comparability, and with the help of this glass framework, AMR surveillance framework, globally data is maintained, data is analyzed, and comparability is there. Every nation, even low income, middle income countries are taking part into it, because WHO's glass AMR framework is we can say like it is not so tedious work, it is not so hard work. Even scientists are generating the data daily, and they can easily put forth the data into this. It is coordinated by nationals laboratories. There is a national antimicrobial resistance monitoring system, and under this NARMS, One Health global framework is that is being conducted, and under this One Health, humans and animals and environment, human health, animal health, and environment health, this is according to that One Health AMR initiative, human health, AMR health, animal health, and environmental health is interconnected. Definitely, it is interconnected, and with the help of this One Health mission, they achieves maximum success from giving the proper guidelines to the communities, from getting AMR surveillance, from getting protected from various easily transmitted pathogens which are highly resistant to the antimicrobial drugs. So we have seen how to access the AMR dashboard. We have seen that how the regional data is there, percentage of data is there, we can easily find it over there. Then NCBI pathogen detection dashboard, it is being coordinated by US National Laboratory of Medicine, NLM, and National Institute of Health. NCBI pathogen detection dashboard. Even on NCBI's dashboard, you can find the pathogen detection dashboard over there, and into that you can find that genomic data into it. That genome sequence data is given in NCBI's portal, and with the help of this genomic data, you can find out the genetic level identification, morphology, sequencing, and which antimicrobial resistant gene is getting inherited regularly. Such kind of data you can find it over there. Hands-on on data retrieval and format harmonization, we have seen that why this data retrieval and format harmonization is important. Because data retrieval is easy, data can be easily retrieved, but format harmonization is one of the important tasks for maintaining the proper standardized records to avoid the confusion of the reader, everything. And then last is the activity metadata integration. Okay, thank you. With here, I would like to thank you. I would like to end up my session here with